Welcome back to the second video reviewing the basics about T cells. In the last video, we introduced one of the core rules of T cell biology. T cells do not usually recognize whole pathogens directly. Instead, they recognize peptides displayed on MHC molecules. But that raises two important questions. First, how does a T cell learn to recognize MHC in the first place? And second, how does the immune system prevent T cells from attacking the animal's own tissues? That is where the thymus comes in. Think of the thymus as the T cell boot camp. This is where developing T cells are tested, trained, selected, and sometimes eliminated. The goal is not to create perfect T cells. The goal is to create T cells that are useful, safe, and ready to participate in immune defense. Hi, I'm Christopher. In this video, we're going to focus on how the thymus shapes T cell population before those cells are allowed to circulate through the body. T cell precursors originate in the bone marrow, then migrate through the bloodstream to the thymus. Once there, they become thymocytes and begin the developmental steps that make them useful, safe, and specific. At this early stage, they are not ready to respond. They still need to develop diverse T cell receptors, pass through CD4 and CD8 stages, and be tested through positive and negative selection. The testing matters because two problems are possible. First, a T cell that cannot properly recognize self-MHC is not useful because it cannot inspect antigen properly. Second, a T cell that recognizes self too strongly is dangerous because it can contribute to autoimmunity. Thus, the thymus is trying to find a balanced middle ground. T cells that can recognize self-MHC but do not strongly attack self-antigens. The first major checkpoint is positive selection. Positive selection asks a simple question. Can this developing T cell recognize self-MHC at all? This mainly happens in the thymic cortex, where developing thymocytes interact with cortical thymic epithelial cells. Those epithelial cells display self-MHC molecules. The developing T cells test their T cell receptors against those MHC molecules. If the T cell receptor binds self-MHC with appropriate low to moderate affinity, the cell receives a survival signal. That survival signal is the positive in positive selection. This process creates MHC restriction. In other words, surviving T cells are restricted to recognizing antigen only when it is presented on the animal's own MHC molecules. That's a big deal because T cells do not just recognize peptide alone, they recognize peptide plus MHC. Positive selection ensures that the T cell can actually use the animal's MHC display system. Now we move to the second major checkpoint, negative selection. Positive selection asks, can you recognize self-MHC? Negative selection asks, do you recognize self too strongly? This step occurs mainly in the thymic medulla. Here, developing T cells encounter self-peptides displayed on self-MHC by medullary thymic epithelial cells and dendritic cells. If a developing T cell binds too strongly to the self-peptide self-MHC, that is a warning sign. A T cell with strong self-reactivity could leave the thymus and attack the animal's own tissues. So instead of being allowed to mature, that cell is eliminated through cell death or apoptosis. This process is also called clonal deletion. Negative selection is one of the central tolerance mechanisms of the immune system. It does not remove every possible self-reactive T cell, but it removes many of the most dangerous ones before they enter circulation. So positive selection builds MHC restriction, negative selection promotes self-tolerance. Together, those two processes help produce T cells that are both functional and safer for the host. Positive selection also helps guide lineage commitment. Developing thymocytes pass through a stage where they express both CD4 and CD8. They are called double positive thymocytes. During positive selection, the type of MHC molecule that the thymocyte recognizes helps determine what kind of T cell it will become. If the thymocyte recognizes self-MHC class 1, it will commit to a CD8 lineage. That cell becomes a CD8 positive cytotoxic T cell. CD8 T cells are important for recognizing infected or abnormal host cells, such as cells presenting endogenous peptides on MHC1. Now, if the thymocyte recognizes self-MHC class 2, it will commit to a CD4 lineage, and that cell becomes a CD4 positive helper T cell. CD4 T cells help coordinate immune responses. They can help B cells produce antibodies, activate macrophages, and shape broader immune response through cytokines. Thus, the thymus is not only asking whether a T cell can recognize MHC, it is also helping determine what kind of job the T cell is going to perform. MHC1 recognition leads to CD8, MHC2 recognition leads to CD4. By the end of thymic selection, we want three major outcomes: restriction, tolerance, and diversity. First, restriction. T cells must be MHC restricted. They need to recognize antigen in the context of self-MHC. Positive selection helps ensure this. Second, tolerance. T cells should not strongly attack self-antigen. Negative selection removes many dangerous self-reactive T cells, reducing the risk of autoimmunity. Third, diversity. The immune system still needs many different T cell receptors. We do not want a tiny identical army of T cells. We want a broad repertoire capable of responding to many future antigens. This is the balancing act of thymic selection. Too little recognition, and the cell is useless. Too much self-reactivity, and the cell is dangerous. Appropriate recognition with enough diversity gives the animal a useful T cell population. So the thymus is not just filtering cells, it is shaping the future immune response. This brings us to the Goldilocks summary. The thymus is looking for T cells that are just right. If a developing T cell is too weak and cannot recognize self-MHC at all, it dies by neglect. If a developing T cell is too strong and binds self-antigen too intensely, it is eliminated during clonal selection. But if the T cell recognizes self-MHC appropriately and does not strongly attack itself, it can survive and mature. That gives us the three outcomes we care about. Restriction. T cells can recognize antigen presented on self-MHC. Tolerance. T cells are less likely to attack self. Diversity. Many different useful T cell receptors remain available to defend against future threats. The thymus does not make perfect T cells. It makes the right ones, strong enough to recognize, restrained enough to avoid major self-reactivity, and diverse enough to protect the host. In the next video, we will build on this foundation by looking more closely at MHC itself, how cells display antigen, how MHC class 1 and MHC class 2 differ, and why those pathways determine whether CD8 or CD4 cells respond. Thanks for watching. I'm Christopher Lee, and what you understand today helps you protect patients tomorrow. And I'll see you in the next video.